Hi everyone, welcome to the urinary system lab. So like the other ones, we're going to have an anatomy and a physiology portion to this one as well. I'll try to say in order this time, and here's the anatomy part. So it is what you've come to expect. There's going to be some key terms, prefixes, suffixes, some microscopic things, and then some larger anatomy. If you click on this link to the lab manual, it will take you to this page. And if you scroll down, there'll be some introductory paragraph that helps explain some of the things to you. Again, the objectives restated. Here are your prefixes, as well as one suffix. And then here are your key terms for you. Again, these will be simple fill-in-the-blank kind of questions on the practical. There is a chart of the blood flow through the kidney. This will help you because some of these terms are very similar to one another. So you will go from the abdominal aorta to the renal artery to the segmental artery, and then this one, interlobar artery to an arcuate artery into an interlobular. Oh, look at that, sneaky, lobular. So this will be one where you have to be really careful when you get to spelling that you are not mixing up these two blood vessels. It will then go to an afferent arteriole, the glomerulus, an efferent arteriole, paratubular capillaries, and vasorecta, which then eventually flow into the interlobular vein, and then we go in the reverse order, and now everything is a vein. So you go from an interlobular vein to an arcuate vein to an interlobar vein to a segmental vein to renal vein to the inferior vena cava. Oh, excuse me. I am going to sneeze. As far as once blood is filtered out of the blood vessel and is caught by the Bowman's capsule or the glomerular capsule, the flow of that fluid or the filtrate goes from that capsule to a proximal convoluted tubule, so it just means it's proximal, meaning it's closest to this capsule, to a descending limb of the Lupa Henle, meaning it goes down, to an ascending limb, which will go back up, to a distal convoluted tubule, because if you have something that's proximal or close to this, and this is obviously further away because it's had all this length to travel, that distal convoluted tubule will empty into a collecting duct, which then merges into a papillary duct, into a minor calyx, major calyx, out the renal pelvis, and then the ureter, which will then take the urine to the bladder. Okay. So again, you'll have your list of structures. We have different models. Scrolling down, we get some microscopic anatomy. We'll have a slide known as the kidney slide. So you need to make sure you know that it is the kidney that you are looking at. And then these are the structures. Similarly, all of these structures can be found in the renal cortex. And again, we have these terms in parentheses to help orient you toward what the question might be. So if the question is like, what organ is this slide of? That would be kidney. What is this structure? It's a renal capsule. What is this region? Renal corpuscle. And then these are also structures, but hopefully that'll help you so that you understand what we're asking for, like a capsule versus region. And again, just kind of the words we'll use when asking questions. The renal medulla, again, is a region. So here, looking at the microscopic anatomy, this part will be the renal capsule, the outermost part. The cortex is closest to the outermost part. This would be a renal corpuscle. Scrolling down, the medulla, you know you're in the medulla. Can you see how it kind of looks fairly uniform? You know you're in the renal medulla. You're missing see this guy? See this renal corpuscle? It's this, I don't know, darker bally looking thing. That's the inner portion of that is the glomerulus. This outer portion is the renal capsule or Bowman's capsule. Those two together form the renal corpuscle. So that those two together are going to be the renal corpuscle. You'll see that in the cortex. You get down to the medulla and you don't see that anymore, right? There's none of those like bally things here. Let's see, if we zoom in, again, so we see glomerulus, Bowman's capsule. You'll see distal and proximal convoluted tubules. I think we have an image where we're zoomed in. It'll be a little bit easier to explain and point them out, but maybe here you can see them too. The main way you're going to tell is, first of all, if you're seeing these glomeruli, again, these ball-like structures, you're in the renal cortex, and you're going to see lots of distal and proximal convoluted tubules. That's what all of these other, clusters are. They're not glands, they're just tubules. Can you see how, like, these distal convoluted tubules, you'll actually see more of the lumen right here. There's more of the lumen here, more of that lumen. You can see, I don't know, it's just like the cells are a little bit narrower, so you see more of the lumen. Whereas in these proximal convoluted tubules, they're much more, thicker cells. So that lumen is very tiny. So like all in here, you don't see much of this lumen. It's not much here. So any of these, again, these kind of thicker cells, I guess, with very little lumen, that's part of the proximal convoluted tubule. These structures here, where you see more of the lumen, will be distal convoluted tubules. Now, one of the differences is whether or not you see it sort of in this lengthwise arrangement or just kind of a circular arrangement this way. These are, they're convoluted, so they kind of squiggle all over the place. So it just depends on how they got sliced as to whether or not you get this longitudinal versus cross-section of it. So they can vary from either this or this longer kind of appearance. All right, so here we're zooming in a little bit. So you can see these proximal convoluted tubules. You just see these kind of clustered areas of kind of pink cells. Those are proximal. Again, the distal ones, you'll see much more of the lumen. They're much clearer in the middle. The cells are not as thick. Here again is that glomerulus. Here's that Bowman's capsule. And here we're zoomed in even more. So again, here you really are starting to see the difference between these proximal convoluted tubules and then the distal ones. Okay, now the ureter is going to take the urine that is formed by the kidneys and take it to the bladder. So the organ is the ureter. The layers are the mucosa muscularis and adventitia. Again, these two things are in the mucosa layer. These are in the muscularis layer. So if we're looking here, so anywhere, this white area right in here, it's kind of, this weird, abstract kind of shape. But that clear area in the middle, that is going to be your lumen. So all of this from the edge of that going inward, this is the mucosa. You'll have these two muscle layers. The innermost is a longitudinal layer, then the circular layer. Any of this darker purple here along the inside of that lumen, that's transitional epithelium, that's a tissue type. And then again, we have this lamina propria, which is this connective tissue that's connecting the epithelial tissue to the muscular tissue. And that's about it for the histology and the lab manual. If I go back and I click on one of the other links that will take me to the labeled images, it'll take us to our usual Google Drive. So that's this one. So that's right here. Again, as you click on these models, let me see if I can, where'd you go? Try that again. Try to adjust my window here of what I'm recording. You'll see we have labeled images, right? So your bladder will have rugae. Those are folds in the bladder. They will expand to allow for more urine to be held until you have to go and pee. This trigone area is just this triangular area, and you'll notice that like the muscles actually orient toward the urethra. In this case, this is a male bladder because of the I guess the addition of the prostate. That's how you know that it would be in a male anatomy. In which case, the urethra going through the prostate is simply the prostatic urethra, right? And that's going to be the tube that's taking the urine from the bladder out the body. So ureters, kidney to bladder, urethra from bladder out the body. Right, so you can see again, kidney, ureter going down to the bladder. And again, this is a male because you'll see some of the male reproductive organs as well. Like the seminal vesicle, the ductus deferens, all that. We get into kidney anatomy. So this is a larger version of the kidney. You'll see some of those blood vessels that I mentioned earlier. So as you click through, we'll have all of those, as well as the layers, the different structures. Here again is that renal corpuscle. So the whole thing together is a renal corpuscle. Separately, it's the glomerulus, which is the blood vessel. Bowman's capsule is just this outer portion. And then on the surface of the glomerulus, so on the surfaces of those capillaries, are these podocytes, or they're called the visceral layer of Bowman's capsule. It helps control the size of the opening, so it helps control what can be filtered by the kidneys. But basically, again, the fluid in these blood vessels gets filtered out these tiny little holes. It's caught by this Bowman's capsule. It then travels down this proximal convoluted tubule, and then it goes and is adjusted along this descending limb of the loop of Henle, the ascending limb, distal convoluted tubule. So it's adjusted all the way through here. Things are being filtered, reabsorbed, until it becomes, it no longer is filtrate, but it's considered urine as it goes into this papillary duct. And then whatever drips out of here is now urine. It is no longer modified. Okay, so again, those are the macroscopic anatomy, and you guys can click through those to help identify any images. If we go to the other link, it will take you to this resource page. Again, you'll have your microscopic anatomy starting up front. So these are just new, or not to say new, they're different images of the same slides. And then the labeled images of the models are very similar to the ones I just showed you. Maybe they're slightly different. So depending on which one you might like a little bit more or less than the other. So there's just a variety of different ways to learn what these different things are. Okay. As always, if you have any questions, please let me know.